It is, well, they're all mucous membranes. Mucous
Well,
membranes is a
the-
broad
the
term.
nasal cavity, but what's the other
The other one is lining the oviduct, right? So let's talk about that real quick. So when we're looking at cilia, we've got two tissues that you guys have learned. The one was lining the respiratory tract, and the other was lining the oviduct or the fallopian tube, right? So two different functions that are kind of similar. One of them is using, like, moving stuff out, and the other one is like cording the oocyte into the uterus. Does that make sense? So I'll show you a little video of cilia, which is kind of cool. So here's cilia in action, both from a lateral view and then from an apical view. It's kind of cool, huh? This is respiratory cilia. And this is from an apical view. You can see the mucus elevator moving it on up. It's more like a mucus moving sidewalk, really. Okay, and does that make sense? Okay, all right, so, and then last but not least, microvilli. The microvilli are the ones that look kind of like a highlighter on the surface. And microvilli is really only found, as far as you've learned thus far, in one tissue. What is it? Non-ciliated simple columnar epithelium. What's the structure-function relationship? What's microvilli do there? Increases surface area for absorption, exactly. So are you hearing how I talk about tissues? I talk about tissues with structure-function relationships. For me, anatomy is completely useless if you only memorize and do not understand anything about the functionality. So you can probably hear how my complex questions will be, right? Based on the way that I'm trying to teach you. You can memorize all you want, but generally memorizers are not retainers. So you actually have to hook it to something that's meaningful in order for you to recall it. And why am I so invested in this? Well, I end up getting a lot of you guys in physiology. And the more anatomy you retain in physiology, the easier my job is to actually tell you anything that's interesting or meaningful. In addition, you're also going to be my medical professionals if I live here and you do too. So I'm invested in you being as smart as possible, really. Okay. All right, so that's that. And now the last thing, although you can't see it in any of these histology slides, it's actually, they're there. They're just not labelable very easily. Okay, so remember sort of the metaphor that we were talking about with epithelial tissue, and we said basically if you've ever tiled the floor, right, we can say that the top surface or the apical surface of the subfloor is kind of equivalent to the apical surface of the connective tissue, right? And then you put on down the adhesive, and the adhesive is a metaphor for what? Basement membrane. And you put down the cells next to one another, and then I told you there would be grout. And so now I'm going to tell you what the grout is. The grout, instead of being an apical surface feature, it's not at the top, it's a lateral surface feature. Yeah? At the sides. So these lateral surface features are how the cells are stuck to one another. And just like there are different types of grout that you can use when tiling, indoor, outdoor, waterproof, malleable, whatever you need, right? There's different types of grouts that you can use between cells, and these are called lateral surface features. So the first kind is right here. It's a little hard to picture what they're trying to show you, but, well, first, before we go any further, can you look at this and tell that they're trying to represent an epithelial tissue? Yeah? Okay, so the one that we're going to look at is this one first. And to me, it looks kind of like a waterproof zipper that is going three-dimensionally and completely zipping the two cells together. This is called a tight junction, and it's a way of sticking two cells together. And the tight junction is about impermeability. So what you want is, you do not want for things to be able to sneak between the cells. So if something is trying to go from here to here, from the superficial surface and go through, I want to make sure I force it to go in through the cell instead of between the cells, okay? And how I do that is I make an impermeable zipper between them called a tight junction. Why would you want a tight junction? Well, I'll give you two different examples of places that there are tight junctions. One of them is in that non-ciliated simple columnar epithelium lining the intestine. When I absorb nutrients, do I want to pull them into the cell so that then I can put them exactly where I want and also get access to them first, right? I get first dibs on glucose, for instance, if I have an intestinal cell, right? Or do I want them to sneak between the cells and then I don't have as much control over where I put them? Does that make sense to you? So if you pull it through the cell, you have a lot more control about where and how much is being transported. that make sense? So you put tight junctions between the cells so it can't sneak, right? And then here's another location, a completely different functionality, which should also make sense to you. Lining the stomach really doesn't do any absorption. Lining the stomach, there are specialized epithelial cells. I haven't taught you this particular type yet because it really only occurs there. But the stomach is special in a couple of different ways. First of all, you knew it was going to have epithelial tissue lining it, right? And you probably know that the stomach fluid is very acidic. Its stomach acid is a pH of about 2. And the pH of about 2 would irritate your skin if it actually got onto the surface of your skin. It would end up causing a lesion if you didn't actually wash it off. So what happens is the epithelial tissue that's lining the stomach is modified so that it can handle that pH. But what do you think about the connective tissue underneath that? Do you think it can handle a pH of 2? How do you keep that fluid from sneaking between those epithelial cells and damaging the connective tissue deep to the epithelial cells? What are these called? They're called tight junctions. They're zipped together with tight junctions. There's actually a clinical manifestation of when you lose those tight junctions and when you lose that intact barrier of the cells because of some kind of infection or injury, it can cause an ulcer, right? So what happens is as soon as you damage the barrier, the stomach acid will do the rest, right? It'll start eating the connective tissue below, right? It can even go all the way through. You can have a perforated ulcer going all the way through. Does that make sense? So tight junctions are really not so much strong, so it doesn't mean that if I pulled on the tissue at the edges like that, it wouldn't break. It means that if I tried to go this way, I have to go through the cells and not between the cells, okay? There is another type of junction, grout, between the cells that gives you strength, and that's this one right here, and it's called a desmosome. And a desmosome is a really strong spot weld that's going 2 directions. So what happens with a desmosome is I really want the cells to be able to hang together despite stretching and twisting. So do you remember learning about the cytoskeleton in 1610, that structural protein framework inside of cell? So I want you to imagine one cell reaching and grabbing onto the cytoskeleton of the cell next to it, and then this cell reaching and grabbing onto the cytoskeleton of this cell. You're going to have a really strong spot weld, right? It's going to be really resistant to stretching and twisting. Think red rover, right? And then the cells will hold together, right? But does that mean that it's impermeable, or can you go right around it if you're trying to move things past it, right? So it's strong, but it's not impermeable, right? Because it's not a 3-dimensional zipper. Does that make sense? That makes sense? Okay, so this is a desmosome. You use this in areas where you need a lot of resistance to stretching and twisting. You have it in the epithelium of your skin because you're going to actually have a lot of stretching and twisting. Great place to think about it is in a woman, the cervix is like the size of a nostril, and you have to stick a watermelon-sized baby out of it, right? And then ever use it again. It better have some freaking desmosomes in it, right? It's got to like hold that tissue together. So anytime you have a lot of stretching and twisting, you need the cells to hold together, you can use desmosomes. Now, cardiac muscle is not an epithelial tissue, but does it surprise you that between the cardiac muscle cells you have desmosomes? Because you're filling and squishing and filling and squishing, and you need them to hold their structural framework. That makes sense? Okay, and then the last type of grout is really corresponds to what you learned in 1610. So remember learning about channel proteins on the surfaces of cells, right? They're small enough to let ions in and out, right? So what I'm going to do is I'm going to take channel proteins from 2 cells right next to one another and stick them together. And this allows for almost instantaneous communication between the cells. So if you want to simplify, I think of tight junctions as being about impermeability, right? Desmosomes as being about strength, and gap junctions as being about communication. So gap junctions allow for instantaneous communication between cells. Why do you want instantaneous communication between cells? Well, interestingly, with those, the respiratory epithelium that was ciliated, right? The ciliated respiratory epithelium is not going to do you a whole hell of a lot of good if all the cells are waving willy-nilly different directions. What I really need them to do is wave in a coordinated wave-like motion up, right? I want them all to be going the same direction. Well, it's ions that start the wave, and the ions start down here and then move here and then move here and then move here and then move here via the gap junctions. And so they respond in the order that they got the stimulus, and then the whole tissue behaves as like it's a single unit because it's connected via gap junctions. Cardiac muscle also has gap junctions. Okay, so what if a tissue needed both strength and impermeability? How would you accomplish that? Which two would you use?
Strength and
Impermeability, right. Desmosomes and tight junctions, right? Or strength and communication, desmosomes and gap junctions. So what I'm saying is there's no limitation, right? You can use more than one type if the tissue has multiple needs. Okay? Okay. So that is your...
with all three?
Huh?
Is there any with all three?
I don't know of one. Doesn't mean there isn't one, but I've never run into one. I've run into them with pairs of all three, orientations of all three. Okay, so gap junctions are the last one. And then the last thing before we move on to like just a little bit about glands and glandular epithelium is, so part of the reason that students have trouble with histology is the 3D versus 2D thing. And then the other part is that they don't really know how to study it. So how do you know when you're finished studying histology? What most people do is they flip through the textbook, they look at that picture, they identify that as simple cuboidal epithelium, and then they go on to the next picture. But what you need to know is the possible questions that you could be asked from any epithelial histology slide. And then if you look at 10 of them, and every single time you can answer all of those questions about 10 different iterations of the same tissue, right? Then you're ready to move on, right? So what are the possible questions? What are the reasonable questions for any epithelial tissue? So if you look at any epithelial tissue, and it doesn't make any difference which one. Let's look at this one. This is a little better. So if we look at this epithelial tissue right here, the possible questions. So first question sometimes throws people. What general category of tissue is it? There's 4 possibilities, right? Epithelium, connective, muscle, and nervous. Don't get thrown by a simple question, right? Which of these categories does it fit into? And then as far as most people ever get is that second question, which is name the epithelial tissue, right? And they go, what is this one? Any idea? Simple columnar epithelium. Yes, that is simple columnar epithelium. Cilia or microvilli? No, those are microvilli. Microvilli. So that's microvilli. All right. So, but modified structures. This is where people get out. So anything that can be labeled there is fair game. So this, name this structure. That's microvilli. You could also, for instance, in another slide, people are not ready for this, but they should be, name this structure. Goblet cell, name this structure, cilia, right? So you need to be prepared for all of those things as well. And then the next one that sometimes people are prepared for and sometimes they're not is name this surface. Apical surface, name this surface. Basal surface, and then assuming you've got a good one, name the structure, basement membrane. So you need to be prepared for that every single time. And of course, like I said, I'm going to use the best slide I can, right? But you need to look at some variability so you get confident with something other than the very, very best slide, right? Especially since your lab instructor may or may not use the best slide every single time. All right, so people are a lot of times prepared for that. This is what people do the most poorly on. Name a specific body location, and you have to be specific, okay? And what do I mean by specific? By specific, you have to play magic school bus and get me to exactly the right place, okay? Like shrink me down, get me to exactly the right place. Like for instance, let's try this one. Name a specific body location for this tissue. Epidermis is good. What if you couldn't remember the word epidermis? Could you not still be descriptive enough that I could play magic school bus with you? Superficial surface of the skin, right? Would you get it correct if you just said skin? You would not, because the skin is composed of more than one tissue, right? So that's not even nitpicky, right? The skin is keratinized stratified squamous epithelium on the top, and then greater or lesser amounts of areolar connective tissue, maybe even fat, and then the dermis is dense irregular connective tissue. So you have to be specific about what you're saying. Okay, so let's practice a couple more. Name a body location for this tissue. Be more specific. Lining the urinary bladder, yes, because the urinary bladder has this tissue, and then connective tissue, and then smooth muscle, and then you're on your rad. Do you see? So what I'm trying to get you to do is to picture where it occurs and where it doesn't in the organ. Okay, let's do a couple more. All right, so let's try this tissue. What? More specific. Lining the fallopian tubes, yes. Okay. I know it sounds nitpicky, but it's actually not true about all portions of the fallopian tube. The fallopian tube has epithelial tissue, connective tissue, and then smooth muscle in the wall, right? So what I'm trying to get you to do is to put it in there, 3D, and then put it in there correctly. And then by the time we get through all the other tissues, you will be able to tell me, oh, okay, I know what I'm going to see if I do a cross-section of the fallopian tube. I'm going to see I'm going to see ciliated simple columnar epithelium lining it, and then I'm going to run into a connective tissue, probably a relar connective tissue, and then I'm going to run into smooth muscle, and then I run into something else on the way out, right? So it gets more important that you're specific once you start building your knowledge, right? Because it doesn't really mean anything if you just say one organ. Is that okay? Okay, so that is what students have to practice, but here's the deal. I only need you to know one very specific, one very correct body location for each tissue, which means that that's going to be a fill-in-the-blank problem, right? Unless, of course, there's only one location for it, right? Like with this one, the only one that I think you learned was lining the fallopian tubes, and with this one, right, it's always lining the urinary tract in some way, shape, or form. But a lot of tissues, there's multiple locations for them, but I need you to know one true one, right? I could have you learn them all, but if I have you learn one true one, then you have to be able to fill it in on a blank, right? Instead of a multiple choice question, because I don't know which one you learned. Good? Okay? All right. So now, and the last thing about this is that don't forget that tissues are simply a bunch of cells. So don't forget that that's a what? Nucleus, and then a cell membrane and cytoplasm. So that stuff is relevant. You can put it together as well. That's not a trick question. Okay? All right. So once you have done all of those with multiple iterations of the same slide, you're golden and you can move on. And if you're really successful at doing it. Also, don't forget the important skill that people don't practice often enough, which is not just identifying, but writing it down. Especially since you don't tend to spell things very well if you haven't practiced writing them. So when you are practicing over and over again, especially once you're confident with your identification, then stop just saying it and start writing it, right? Especially since in lab they count off a decent amount up to a letter grade on a test for spelling. Okay, I'm a little more gentle about spelling, and except for the fact that if you spell it badly enough, it's a different word, right? So I try to give you the benefit of the doubt. Okay. So the last thing about epithelial tissue is simply glands, and we have to totally switch gears when we're talking about glandular epithelium. So like we've been talking about before, epithelial tissue tends to have a free surface. That's what we were saying, right? And then it's going to have connective tissue underneath it. But glandular epithelium doesn't necessarily adhere to these rules. It just happens to be made of epithelial tissue as well, meaning that even though the pituitary gland is deep, just inferior to my brain in the middle of my head and nowhere near a surface, I'm telling you that glands are made of epithelium. So the pancreas is made of epithelium. Mammary glands are made of epithelium. Sweat glands and oil glands and your pituitary and your thyroid, all of that is made of epithelial tissue, even if it's not close to a surface. Now, the reason that I don't usually ask you to identify histology slides of glandular epithelium is because they look like a big freaking mess and they're really hard to do. But I do want you to understand that glands are made of epithelium, and that's clinically important for this reason. Remember what I told you about the cell division and regeneration rate of epithelial tissues? Fast or slow? Fast. Glands also have a fast rate of cell division and regeneration, and that is clinically important because cancer is uncontrolled mitosis or cell division, when you lose control of mitosis. Does it make sense to you that the tissues that are most likely to have uncontrolled mitosis are the tissues that have the highest rate of mitosis, right? because it's just statistics, right? Can you lose control of it if it happens very, very frequently? So then broaden your view and think about the cancers that you hear about most frequently. They are almost invariably epithelial cancers. You've got skin cancer, you've got lung cancer, which occurs initially in the lining of the lung. You've got thyroid cancer and pituitary cancer. You've got breast cancer, right? Does that make sense to you guys? So the reason that it's clinically important that these fit into the category of epithelial tissues is that it tells you something about their predisposition. Does that make sense? So there are other cancers, they just don't occur as frequently. Like, for instance, the cardiac muscle, in adulthood it's amitotic. It doesn't undergo cell division normally. And that probably corresponds directly to how frequently you've heard of heart cancer, right? And I'm not saying it doesn't occur, because anything that can go wrong will go wrong eventually, but you certainly don't hear of it frequently, right? So the rate of cell division can tell you something about the rate of losing control of cell division. If you also combine that with blasting an epithelial tissue with known mutagens or carcinogens like UV radiation or toxins, then you're increasing dramatically the likelihood that that epithelial tissue will become cancerous. So remember that as you decide to put on sunscreen in the morning. It's like epithelial tissue and a known mutagen and carcinogen. So, okay, so let's look at some examples of glands. A gland is simply a cell or more than one cell that produces and secretes a product. And a lot of your glands are just deep to, so they call the typical epithelium, they call it covering and lining epithelium. Many glands are just deep to that and dump their products onto the surfaces, but not all glands, right? So a pituitary gland doesn't fit that criteria, but a salivary gland does, and a sweat gland or an oil gland does. They're just deep to the covering and lining and dump it onto the surface. So there's actually two big categories of glands depending on where they dump their products. An exocrine gland dumps its product onto the surface of a covering or lining epithelium. It doesn't have to be outside the body, because for instance it could be into the digestive tract or the respiratory tract, right? But it does have to be onto the surface. And then an endocrine gland dumps its products into the bloodstream. Does that make sense? Exo, endo, right? All right, so now I'm going to name some glands for you, and I want you to try to categorize them as exocrine or endocrine based on that categorization. So exocrine onto the surface of recovering the lining epithelium, and endocrine into the bloodstream. So pituitary gland, endocrine, salivary gland, exocrine, thyroid gland, endocrine, mammary gland. Mammary gland is exocrine, onto the surface of covering the lining epithelium, right? Does that make sense? Goblet cell, exocrine, right? So I'm not saying it goes outside the body, I'm saying it goes onto the surface, right? And endocrine goes into the bloodstream. So the endocrine secretes hormones, right, into the bloodstream. And the exocrine secretes multiple different kinds of products, but always onto a surface. Does that make sense? Okay, so exocrine glands, we already met the unicellular exocrine gland, which was a goblet cell. And that's this guy right here. I'll remind you of what it looks like. So do you see that that fits the criteria of dumping its product onto the surface of a covering or lining epithelium? That one just happens to be unicellular. And then once you get more complex exocrine glands, so I'm not going to have you look at these and identify them by name, but you should be able to look at them and tell that they're epithelium and that they're exocrine. So these are a bunch of different exocrine glands, but notice that what they are is just deep to a covering and lining epithelium. Yeah, do you see that? No matter how branched they are, they're just deep to a covering and lining epithelium. And then if you look at the structure, doesn't it actually look like an epithelial tissue? Just like a super curvy epithelial tissue. So what happens with the multicellular exocrine glands is that they tend to be deep to a covering and lining epithelium, and then they may or may not be really branched, but they're always going to have a tube that leads up to the top, right? And that tube is called a duct, right? And if you look at it from the top, it looks like a pore maybe, right? That makes sense? But endocrine glands are ductless because they don't go to the surfaces of things, right? They don't have a duct that leads to the surface. That makes sense? So like pituitary and thyroid and all that kind of stuff. So that's multicellular exocrine glands. So again, you don't have to recognize them individually, but you should recognize that they're epithelium and that they're exocrine, right? And then endocrine, oh, I lost my place. Endocrine are ductless and they secrete hormones, and you can think of tons of examples of endocrine glands. Anything that secretes a hormone, including the pancreas and the thyroid and the pituitary. We don't do a whole lot of endocrine anatomy because the anatomy is not particularly interesting, because all they are is they are like completely separate from one another but have a really rich capillary supply because they dump stuff into the capillaries. But you do tons of endocrinology and physiology, tons of it. Okay, so now we are going to change gears and move to connective tissue. And connective tissue, from my perspective, maybe you agree, maybe you don't, it's the hardest of the four. Epithelial tissue is easy to recognize as epithelial tissue once you get used to looking at it. And then it has a naming convention that works at least most of the time. Connective tissue is highly variable in its structure. So you've got blood, fat, ligaments, and bone that are all connective tissue, and they're really variable. And then nobody ever decided on a naming convention. So the naming is really not very consistent either. So to me, it's the hardest one, which means that organization of your knowledge of connective tissue is going to be really important. So before we get into the nitty-gritty of the organization, let's just talk about some variability in connective tissue. The most common tissue in the body, it's the most diverse tissue in the body. It's almost everywhere in the body with the exception of the fact that it shouldn't be exposed to the outside unless something's broken, right? It's usually pretty vascular, especially when you compare it to epithelial tissue, which is avascular, right? Cartilage is really not very vascular at all, but the rest of the connective tissues are. And one of the most easily identifiable things about connective tissue is that you're just going to have cell, and then stuff between the cells, right? So unlike epithelial tissue where the cells are smacked right up against one another, you're going to have cell, stuff between the cells. So let me just show you a couple of views, and you should have started looking at this. So can you see that? A bunch of cells with stuff between the cells. Okay. How about this one? Bunch of cells with stuff between the cells, right? So once you get used to looking at connective tissue, you will be able to look at it and say, I may not know exactly what it is, but I think it's a connective tissue, right? Just like you can with epithelial tissues. So widely scattered cells with lots of space. Go ahead.
Would they always be lines going lateral and parallel?
No, like with blood, there isn't any fibers between them. There's just a lot of fluid between them. So there's still space between the cells, but you can't depend on there being fibers. So we'll talk about that in just a second. Okay, so as far as categories of connective tissue, why conceptually are tissues as diverse as blood, bone, fat all considered the same category? And the answer is two things. First, logically, they don't fit into any of the other categories, right? Epithelial tissue is not on the surface, right? Nervous tissue, no. Muscular tissue doesn't even wiggle, right? So everything else is connective tissue, but really the reason that they're named the way that they are is that they are all derived from the same embryonic tissue. So during the first stages of differentiation, when you go from a zygote to multicelled, then there is an initial organization stage in which you make a layer of tissue, and one of the layers of tissue is going to be the basis for all connective tissues, and it's called mesenchyme. So let me show you a little bit about mesenchyme. Okay, so we're going to come back to this in just a second. So why are they all considered connective tissues? Because they're all derived from the same embryonic connective tissue. And mesenchyme is nothing impressive to look at, right? You could look at it and you're like, I never asked this slide because it looks like nothing. I mean, you can tell it's not epithelial tissue, right? By looking at it. But other than that, they say those are fibers. I'm just going to have to trust them because they don't look like a damn thing to me. I see a bunch of cells with spaces between them. So this is your embryonic connective tissue. This is the connective tissue from which all other ones are derived. So all of the adult connective tissues are derived from that. So We're going to organize them. There's 3 big categories of adult connective tissue that are all derived from that, and we're going to organize them today. One of them is the supporting connective tissues. Those are all the things that help you resist gravity, including cartilage and bone. Yeah? There's the fluid connective tissues. That one's really easy because it only has one tissue in it, and that's blood. And then there's all the other ones, and there are 6 of those little buggers. And those are the connective tissues proper. And they are between fluid and solid. They're kind of squishy, like fat and ligaments and that kind of stuff, right? So those all fit in connective tissue proper. Those are the hard ones because there's 6 of them and you have to sort of separate them from one another. So because connective tissue is so broad in its structure, it is just as broad in its function. So this is just some of the things that connective tissue could do. It's protected in some places, like bone and fat can be protective, or they can help you resist gravity, like bone and cartilage. Or they can strap something to something else, like a ligament or a tendon, or they can store fat. And this is just like a partial list. They do any number of things. They do everything that the other 3 types of tissues do not do. So that's not a comprehensive list at all. So first, I want to organize the connective tissues, and then we will get into like the structure and function of the connective tissues. So you don't have to recreate this unless you want to, but I tend to remember things better when I recreate it myself. So if we're dealing with the adult connective tissues, they're all derived from this embryonic one, which isn't present any longer in an adult, although some of the cells are still around. So the mesenchyme is that embryonic connective tissue. And then mesenchyme gives rise to 3 separate categories of adult connective tissues. And one of them is the connective tissues proper. And those are the squishy ones, not fluid, not solid. Okay? They will include ligaments and tendons and fat and a bunch of other stuff. Okay? And then these are the supporting ones. Supporting connective tissues, and they will include bone and cartilage, okay? And then the easy one, and the easy one is the fluid connective tissues, and that is just blood, okay? So only fluid connective tissue that you're going to learn. So what we're going to have to do is we're going to learn how many fit into this category, that category, and that category. And then we're going to look at examples of each. And then it's going to be fair game not for me to just show you a connective tissue figure, but for me to show you a connective tissue figure and then ask you where it fits. Is it proper? Is it supporting? So you're going to have to sort of figure out where it fits into the story as well. Okay, so blood is done. There's only one of those. That's super easy. The next easiest one is supporting, because there's 5 types of supporting. And they're not super hard to identify one from the other. So there is bone, and there's 2 types of bone, compact and spongy. But you actually don't learn the spongy bone slide until we do bone. So really you only have to know compact for right now. And then there's 3 types of cartilage. And you should be learning these this week, right, in lab? Okay, and there's hyaline and hyaline cartilage, and there is elastic cartilage, and then there is fibrocartilage. And then the hard one is over here. Connective tissue proper is the hard one. And connective tissue proper has a big subdivision under it, and I'm going to show you why it has a subdivision, and then we will do these first. The connective tissues proper are subdivided into the loose ones and the dense ones. So let me show you 2 connective tissues proper, and you tell me which one's loose and which one's dense, and it's not that bad. Loose versus dense refers to how packed the matrix is between the cells with fibers. Okay, so here's A, okay, and here's B, dense, right? So you categorize them as loose versus dense, and there's going to be 3 loose ones, and there's going to be 3 dense ones, okay? So just so that we can be finished with this, and then we'll just find examples of everything, With loose, there is areolar connective tissue and adipose connective tissue and connective tissue. And then with dense, there's dense regular, dense irregular, and elastic. Sometimes elastic is called elastic dense. Sometimes they leave that word out. Okay. So, I mean, like I said, you didn't really have to recreate that because it's right here. But it's nice to be able to do it, and then you figure out where everything fits into the story that you're telling. Okay, it's the same thing that we're doing here. Okay, so when you're looking at a connective tissue, there's a couple of levels of difficulty. First of all, they look very different from one another. There's no naming convention, and then there's multiple things to know about each tissue. The cells will have individual names, and then there will be different kinds of fibers in the tissue as well, okay? So that's kind of your challenge that you're trying to deal with here. So first, let's talk about what the cells could be called in a connective tissue. So when you're looking at cells in a connective tissue, or really anywhere, generally the cells will have one of three endings. It can either be a blast cell, a site cell, or a clast cell. And a blast cell is going to be a precursor to something. It's going to be a really active cell in either making matrix or doing cell division. And so, for instance, in bone, once you look more closely at bone, you're going to run into a cell called an osteoblast cell. And an osteoblast cell will be responsible for making bone matrix, right? Okay. And then when you're looking at connective tissues, you're also going to run into fibroblast cells. What do you think fibroblast cells do? Make fibers, right? And so not surprisingly, you're going to see a lot of fibroblast cells in connective tissues because there's lots of fibers in connective tissues. Is that okay? And then you can run into some other kinds of blast cells in other types of tissues. The more common type of cell that you're going to run into is a little less active in division, a little more mature cell. It's called a site cell, right? And so in bone, it would be called an osteocyte cell. In cartilage, it might be called a chondrocyte cell, and in fat, it might be called a adipocyte cell. Does that make sense? Okay. All right. And then last but not least, you probably won't in this chapter, but you will later in the semester, sometimes run into clast cells. And clast cells are not responsible for building the tissue. They're responsible for breaking down the tissue, usually for remodeling, right? So sometimes if you need to change the shape of something, you don't just need to make more on top of it. You need to do destruction before you do construction. So for instance, you can run into osteoclast cells, which would be responsible for changing the shape of bone, okay? And then anything that is not the cell, so here we go with the cells. Oh, wait, one more thing. There's one other type of cell that seems kind of counterintuitive that you can run into in connective tissue. And if it doesn't end with blast or site or clast, you also can run into white blood cells that wander into connective tissue. And I don't know if you knew this before now, but okay, I want you to think about your epithelial tissues, right, are on the surfaces of things, and then immediately deep to that is going to be connective tissue, right? And remember that in epithelial tissues, there really isn't any room for white blood cells, right? They're completely crowded. So does it surprise you at all that very often white blood cells will move into the connective tissue immediately deep to an epithelial tissue? They'll sort of take up residence there like a sentinel, because epithelial tissues are really the most common place to get exposed to pathogens, right? So white blood cells wander out of the bloodstream and take up residence in the connective tissues. So don't ever be surprised if, for instance, you see this big honker, which is a macrophage that just happens to move in to a connective tissue, right? So I don't know if you knew that before. Red blood cells should stay sequestered inside your cardiovascular system, but white blood cells don't have to. In addition to circulating in the cardiovascular system, they also move into tissues, because then you can launch an immune system response from a tissue or the bloodstream. Does that make sense? Okay, so you're going to run into those not infrequently, and they're called macrophages. Okay, so what else is in there besides cells? Well, when you're looking, and this is just a stylized diagram of a connective tissue, you got cells, and then you've got fibers, right? And we'll name the fibers in just a second. And you have the stuff that is not cells or fibers. And the stuff that is not cells or fibers is generally referred to as ground substance, but that's a super general term. It's actually not telling you anything. So ground substance is in a connective tissue, but not cells and not fibers. So if you're talking about blood, then ground substance is about 90% water. But if you're talking about bone, then the ground substance is mostly calcium crystals, right? So what it's telling you is it is part of the tissue, but it's not the fibers and it's not the cells. And then to know what it's more like, you have to talk about a specific tissue, right? Does that make sense? Okay. So basically, in order to make a connective tissue, you need all three of those things, potentially, the ground substance, the fibers, and the cells. Okay, so now let's talk about the fibers. So in order to make connective tissue connective, quite often it has long, skinny, insoluble fibers, meaning they're not going to break down in fluid. And think of them as, I don't know, part of a fabric, right? The fiber parts of a fabric. And you can use different types to make the fabric have different characteristics. So there's three types of fibers that are commonly used in connective tissue. One of them is called a collagen fiber. And collagen fibers, I think of them as rope fibers, you know, the fibers that you would use to make a rope. Individually, not very strong, but when you start to bundle them, don't they get stronger and stronger, right? Okay. So if you bundled them all parallel, then you would only be strong this direction. But you can also use rope fibers to make a hammock, and they'll be strong a bunch of different directions. So collagen tends to be long and skinny, relatively thick, not as thick as this, which is a capillary, but relatively thick, and it doesn't branch if you follow it. And usually, but not always, it stains kind of bubblegum pink. Can you see the collagen fiber there? Okay, so that's collagen. Elastic fibers are, think of like one of those rubber bands that you use on your braces, like individually very, very thin, right, but stretchy. But couldn't you get strength if you bundled a bunch of them together as well, right? You could get both strength and elasticity if you had bundles and bundles and bundles of rubber bands. So that's what an elastic fiber is. It is thinner. Quite often they branch, okay? Individually, they're not very strong, but collectively you can get some strength out of them. And usually, but not always, they stain much darker than collagen fibers. And usually almost black or dark purple or sometimes blue. So here they are. Can you see the elastic fibers now? Yeah? Much thinner, more likely to be branched. Okay. And then last but not least are these fibers, which are usually so thin that you can't see them in most slides, even if they're present. These are called reticular fibers. And for a metaphor for reticular fibers, I want you to think about a hairnet, right? The fibers are very, very thin, and they're branched, and they form a net. Really hard to see. They don't give much structural stability, but they can hold the shape of a soft organ, for instance. Okay? But like I said, even if they're present, they're not very easy to see. So they are present in this tissue, but you would never have to label it because you can't really identify them. The only tissue in which you have to identify them visually is a tissue that's called reticular connective tissue. And all of the fibers in it are reticular fibers. Does that make sense to you guys? Okay. Now, I'm not saying it's not present in a bunch more tissues, but you don't have to identify it from looking at it. Okay. All right. So going back to your fibers for just a second. Collagen is the most abundant protein in the body. You've got about 30,000 to 60,000. They're still trying to get a good count on how many different kinds of proteins you have in your body. But 25 to 46% of them are collagen. So it's by far the most abundant protein that you have in your body. And its primary job is those rope fibers, right? It's helping you to hold things together and to resist gravity, right? So it's sort of holding you up. Collagen is really like sort of the culprit in the aging process. So what happens with aging is everything used to be way up top, right? And then the persistence of gravity in combination with the fact that you don't repair your collagen fibers as readily as you age Your cell division and protein synthesis processes will slow down as you age. So you break down collagen fibers and you don't regenerate them. And then you start having wrinkles, right? And things head south, right? Up top, down low, gentlemen, don't think we don't notice. Everything just heads south. So the sort of progressive breakdown of collagen is part of that. You also lose elastic fibers as you age. If you snap your kid's skin, you know, you got a little kid, their skin snaps back immediately. But if you do it to yours when you're in your 40s, it's like, well, I'll get there eventually. Maybe. Okay? I'll eventually do that. So your collagen and your elastic fibers both start to degenerate as you age. But I will tell you that there are some things, habits, behaviors, that you can do which will make them break down way, way faster. Does anybody know a couple of different things that dependably break down collagen and elastic fibers? Smoking. Yes, smoking is absolutely horrendous for your collagen and elastic fibers. It breaks them down and they have trouble regenerating. And so I'm not from Utah, and people all over the South smoke a lot more than they do in Utah. And I put myself through college working in restaurants. And the first question when people come in would be smoking or non-smoking. But if you're a woman, I don't even have to ask you if you smoke. All I have to do is look at your skin and I can tell whether you smoke. So it's immediately obvious whether somebody smokes. The other thing, which they do a lot more of here in Utah, there's one other thing that dependably breaks down your collagen elastic fibers. And what's that? Yes, sun exposure. UV radiation breaks down your collagen and elastic fibers and will age your skin like crazy. So just remember the hot that you're going for at 22 is going to cost you when you're 40, right? And just remember, everybody's pretty hot at 22. It's kind of the default, right? Way harder at 40, just way more work, gotta tell you. Okay, so that's collagen, elastin, and reticular. And then, so our types of connective tissues, we'll do this in more detail next time, but let me just tell you last thing about mesenchyme. So mesenchyme was your embryonic connective tissue from which all other connective tissues are derived. As an adult, you don't keep mesenchyme, but you do keep mesenchymal cells. And the cool thing about mesenchymal cells, they're just sort of sprinkled around. You can see them right here. See that mesenchymal cell? They remember how to make any connective tissue. So they were embryonic connective tissues from which all other connective tissues are derived. So for instance, if you damage something and have trouble with regeneration, mesenchymal cells will do their dead level best to help you regenerate. You need some bone, I'll make some bone. You need some cartilage, I'll make some cartilage. Now, they have limited capacity because there aren't that many of them. But sometimes that's the only connective tissue regeneration that you get. Okay? So I will see you guys on Tuesday. No quiz on Tuesday.